Thanks to host for having me in this webinar. My name is Ivan Cohen, and today I'm going to present on in vitro immunogenicity prediction. I will specify my talk on effective methods in drug development to identify potential unwanted immune response beforehand. Immunogenicity can have different meanings in the industry. When I refer to immunogenicity of biotherapeutics, I refer to it when it occurs when the immune system develops anti-drug antibodies against biotherapeutic, which is an unwanted immune response. In this slide, there are several examples of how immunogenicity had substantial consequences on drug development, leading to reduction in safety or efficacy of the biotherapeutic and causing, in some cases, early termination of this molecule. And as such, immunogenicity may be a big problem for the industry. But what if we were able to assess immunogenicity risk preclinically? And this is exactly what my group in Genentech is doing. We are developing different methods to assess immunogenicity risk of biotherapeutics before clinical trials. This we are doing with different methods such as in silico analysis or T-cell activation assay. And today, I'm going to talk about the in vitro methods that can be used in order to assess immunogenicity risk. Before I dive into the methods themselves, let me first talk about a bit of a background on how immunogenicity occurs. So the three different key players in immunogenicity, the major players, are the antigen-presenting cells, which uptake the drug and present it to the T cell. The T cells are the ones that will determine whether there will be a continuum of the signal interacting with B cells, leading for their differentiation into plasma cells that will secrete the anti-drug antibodies. And now that we know the key players, let me go into a bit more details on how this process occurs. Of course, this is a very complex process, but I will try to summarize this in this slide. So when a drug enters our body, it's being uptaken by antigen-presenting cells. In the antigen-presenting cells, the drug will be cleaved and processed to small peptides. Those peptides can bind, in some cases, if they have high binding affinity, to the HLA class II. And then, in some cases, they can be presented. If there will be a T cell that recognizes this complex of HLA class II and a peptide, this T cell can become activate, differentiate, and proliferate, and later on interact with a B cell. The B cells on their end can also uptake the biotherapeutic and process it, present it on the HLA class II to this activated T cell. This interaction of T cell and B cell may lead for differentiation of the B cell to a memory cell or to a plasma cell that will secrete those anti-drug antibodies. And why are those anti-drug antibodies are so detrimental? Because they can alter pharmacokinetics in some cases, reduce efficacy in other cases, and they can also, in some rare cases, impact safety. For example, they can interact with endogenous counterpart of the molecule, the biotherapeutic, that share the same sequence, leading to autoimmunity. Of course, we always need to remember that those ADA might be not effective and might not lead for those consequences. But when we are thinking about immunogenicity, we need to always think about the worst case scenario. Now that we understand how immunogenicity occurs, let me talk a bit about the factors that influence immunogenicity. Those factors are important to understand when setting those immunogenicity prediction in vitro methods and comparing them to the clinical outcome of different benchmark molecules. But they are also important to give final assessment of what will be the immunogenicity risk of the tested molecule. We need to understand those factors when developing the different assays, so we will be able to fit the chosen method or the chosen in vitro assay to the factors that we are investigating that may influence the immunogenicity of our molecules. Those factors can generally be divided into two categories. One, the product-related factors, and two, the patient-related factors. In the product-related factors, we can find factors such as the sequence of amino acids that our biotherapeutic is comprised of. Those amino acids may form peptides with high binding affinity to the HLA class II, for example. This may increase their chances to be presented by multiple HLA class II, and by that increases their chances to be more immunogenic. Second, in our factors that influence immunogenicity, we can find the biotherapeutic itself, whether it can form aggregation. Those aggregations may increase our immunogenicity risk, and those may not be detected if we will only examine the sequence of our molecule by in silico algorithms, for example. Also, our product may contain impurities such as O-cell proteins. The foreignness of those O-cell proteins may increase the chances of this molecule to cause a high ADA in the clinic. The MOA of the molecule may also play a significant role in determining its clinical ADA. For example, if the molecule increases or decreases the immune response. Some biotherapeutics, for example, that completely deplete CD20 may not show high anti-drug antibodies in the clinic due to the depletion of B cells, but may still appear as highly immunogenic by in vitro or in silico tools. In the patient-related factors that influence immunogenicity, we can find co-medication, for example. Immune suppressors may depress the entire immune system, and the percent clinical ADA that we will observe in these patients may be lower than patients that do not take those immune suppressor drugs. The patient's own immune system and whether it is activated or suppressed may also influence our ADA. And the patient's own genotype, for example, its HLA class II, may also indicate if some biotherapeutic may have higher chances to be immunogenic in this patient. Also, the dose and route of injection of the biotherapeutic may be important, as we expect that multiple dosing and subcutaneous injection may increase in some cases the generation of ADA in some patients. Lastly, as those immunogenicity predictions are typically developed based on a set of known molecules that their clinical ADA is known, it is really important to understand that one of the most crucial factors that will determine clinical ADA is how this clinical ADA is measured and how the assay is set up to evaluate the clinical ADA. Now that we understand why immunogenicity is so important to determine before clinical trials and how it occurs and what are the factors that influence immunogenicity, let's look at what the regulatory guidance on immunogenicity. Let's think about it. So they typically encourage those use of those assays, but this is not a mandatory. As it says in the guidance, non-clinical in vitro and or in vivo studies aiming at predicting immunogenicity in humans are normally not required. However, ongoing consideration should be given to the use of emerging technologies such as novel in silico, in vitro, or in vivo methods, which might be used as tools during development or for a first estimation risk of the clinical immunogenicity. And this is what we are doing. We are trying to develop those tools to give us an assessment of the risk of immunogenicity. Based on what we know on how our immune response leads to immunogenicity of biotherapeutics, different methods were developed to try to mimic this process in vitro, and by that, estimate the immunogenicity risk of the different drugs. As the process leading to immunogenicity is complex and involves multiple stages, we always need to remember that we can use those methods to assess the immunogenicity risk. But the final outcome of how our biotherapeutic and what is the exact clinical ADA it leads to can only be truly determined once we inject this to the patient. The different in vitro methods to assess immunogenicity risk are addressing different stages of the immune response. And as we move more further in those steps, the immunogenicity risk may capture more factors and maybe be more predictive. The first step, where the biotherapeutic enters our body, leading to the initial step, triggering immune response, can be assessed by, for example, DC activation or drug internalization. Those assays aim to capture how well is our biotherapeutic in uptaking and sometimes degrading by the antigen-presenting cells, and whether those biotherapeutic will activate our innate immune response. The next step in the immune system against the biotherapeutic would require that the biotherapeutic or peptides of this biotherapeutic be presented by the antigen-presenting cell. This step can be assessed, for example, by in silico algorithms. The current in silico algorithms are aimed to predict, based on trained data sets of peptides, what are the areas within our biotherapeutic or for a given peptide that will bind or be presented by the HLA class II. By proxy, increased binding or presentation of those sequences may suggest increased immunogenicity of the drug. The other method is in vitro HLA-2 binding. In those assays, we test in vitro whether a specific peptide can bind to a specific HLA class II. Another experimentally assay to assess exactly what are the peptides within our biotherapeutic that are being presented by the HLA class II is MHC protein proteomics, or MAPS, which I will elaborate a bit more in a couple of slides. In most cases, in order for our immune response to develop anti-drug antibodies against our drug, T-cell-dependent immune response must occur, and our T-cell will have to recognize the peptide of the biotherapeutic that is presented by the HLA class II. This process will trigger T-cell activation, proliferation, and cytokine secretion. And the next step of methods are aimed to evaluate this response. And again, I will elaborate on this in a few slides. Lastly, in some cases, the presence of pre-existing antibody may be detected by immunoassays that will be pre-screened for the patient. For example, for a pegylated molecule, we may find pre-existing anti-drug antibodies even without administering the biotherapeutic. Now that we went over the different methods more generally, let's dive more into the specific. As the first step leading to immunogenicity would occur when the drug is internalized into the antigen-presenting cell, two different sets of experiments may be used to further evaluate this step and its consequences. One, DC activation, and two, assay that specifically evaluate drug internalization. For DC activation, there are multiple outputs that the assay may have and be developed to test. For example, in a cross-industry study done by the ABRES group, different outputs were used to estimate the immunogenicity risk of aggregated biotherapeutic. It was shown that all the methods used to assess activation of the innate system, such as RT-PCR, cytokine secretion by Luminex, and flow cytometry, achieved the same goal, showing that aggregated molecules may be more immunogenic. Drug internalization can be looked by different methods to evaluate by flow cytometry or microscopy. Several groups have shown a correlation between the amount of drug internalized and degraded with immunogenicity risk. Molecules with high clinical ADA have shown to be highly internalized by the DC, and the opposite was shown for molecules with low clinical ADA. To identify the immunogenic sequence within our biotherapeutic, we can go with different approaches. For example, the in silico algorithms that try to assess if a certain peptide can bind or expressed by the HLA class II. A second method, MAPS, is experimentally looking at what are those exact peptides that are being presented by the HLA class II. However, to understand this method, we need to better understand the complexity of the HLA class II molecule. With MAPS, we can identify what are the peptides that are being expressed by the HLA class II. However, we need to understand that the HLA class II is comprised by three different proteins, HLA-DR, HLA-DP, and HLA-DQ. Each one of these molecules is comprised by two different chains, alpha and beta chain. And in the case of HLA-DR beta, HLA-DP alpha and beta, and HLA-DQ alpha and beta, those are highly polymorphic genes. And both the alpha and beta chain may impact the binding of the certain peptide to the HLA class II. Meaning that for a certain peptide, we might see different HLA class II being presented on the cell surface and presenting different peptides of our biotherapeutic. In MultiMACS assay, we are looking at different peptides that are being presented on our antigen-presenting cells. Typically, experimentally, those will be monocyte-derived DCs. The process to generate those cells will require first isolation of CD14 cells from BBMCs, and then culturing them for a few days with a cocktail of cytokines, and then maturing them and adding of the biotherapeutics. Later on in the MultiMACS process, we will IP the HLA-DR and elute the peptides, and then run them by mass spectrometry to see the different peptides that are being presented. Those peptides that are identified by the mass spec can visually later on be aligned to the molecule itself or the biotherapeutic, in this case it's an antibody, generating an heat map that will show us exactly what are the areas that are being expressed of this biotherapeutic. As you can see in this scheme, multiple donors have been tested. This is because different donors or different PBMCs from donors have different HLA class II. Different HLA class II can have, as discussed before, different binding and expression of the certain peptides. And therefore, we need multiple donors when we are running this assay. You can see here that there is an area between the CDR1 and the framework 2 that might be promiscuous as it is presented by multiple donors. Also can be seen here that there is an area in the construct domain that is being presented. However, since this area in the construct domain also exists in endogenous antibodies, this may be tolerized and won't cause an immunogenic risk. This is why when we are running MAPS, we always need to run this in conjugation with multiple donors first, and also in conjugation with other assays, such as the T-cell activation or T-cell proliferation assays that I'm going to talk next. Even if a peptide from the biotherapeutic is being presented by the HLA class II, it won't guarantee that there will be an immune response. Only if there will be a T-cell with a specific TCR that recognizes the peptide HLA class II complex, there will be a T-cell response. The next set of methods are aiming to assess T-cell response. Those are done by evaluating T-cell activation, cytokine secretion, or T-cell proliferation. One of the most common outputs to evaluate T-cell response due to biotherapeutic in vitro and estimate by this the immunogenicity risk of the biotherapeutic is looking at T-cell proliferation assay. The assay can be set to look at different outputs, such as incorporation of H3 thymidine, or by flow cytometry to look at BRDU incorporation, or by different markers for staining markers, such as cell trace violet, CFSC, or proliferation markers, such as Ki67. Those will evaluate proliferation of the cells. By conducting this assay in a flow cytometry, we can gauge on the desired population. In case of immunogenicity risk, we are interested to look at the proliferation of T helper cells or CD4 class population. So how do we typically set those experiments? Those will require incubation of PBMCs or CD4 plus enriched PBMCs. Those cells are depleted from CD8, but we keep the other PBMCs during the culture because we need the antigen-presenting cells to present the peptides of the biotherapeutic. This step enables a better identification of those rare antigen-specific or biotherapeutic-specific CD4 proliferation cells. This incubation can be as long as 6 to 8 days. We are trying to get those proliferation cells to be evaluated. We can also re-stimulate the cells over and over with a biotherapeutic if needed. In this study that I'm going to present you next, we use BRDU to track the proliferation of T helper cells in our culture following stimulation with multiple biotherapeutics. In this case study, to show you the capability of T-cell proliferation assays to assess immunogenicity risk of biotherapeutics, we have used multiple biotherapeutics. Each one of the PBMCs were stimulated with one of these biotherapeutics. Bevacizumab, this is a low immunogenic biotherapeutic with 0.6% clinical ADA. Also, we stimulated the cells with a panel of anti-PCSK9 antibodies. Alirocumab, which is a low immunogenic PCSK9. Avolocumab, this is another low immunogenic anti-PCSK9. RG7652, this is Genentech's own anti-PCSK9 that showed to be low immunogenic in the clinic. And we also stimulated with another molecule, bocukizumab. This molecule showed to be 48% clinical ADA in phase 3 and was terminated. Since all of those molecules share the same target, we thought it would be nice to compare between them and see if we can be able to assess this by the T-cell proliferation. Another highly immunogenic biotherapeutic that we have tested in this study was HA33. This is a molecule that showed 73% ADA, which is considered very high. In the studies, we also used a strong antigen, such as KLH, to be our positive control. We expect a CD4 proliferation when we stimulate PBMCs with this. As you can see here on the y-axis, we have the positive percent of positive donors. I can see here that it's a bit shift, but the 100% is the top, which is what we got with KLH. And you can see on the x-axis, those are the biotherapeutics. We also summarize the percent of clinical ADA again to remind you of what's the clinical ADA, and the percent of positive donors on the bottom part of the table that shows you our outcomes from the analysis. You can see here very clearly that the highly immunogenic biotherapeutics that we have tested, bocukizumab and HA33, were shown to be more immunogenic or leading to more positive donors than the other biotherapeutics. Bocukizumab was much higher than the other PCSK9 antibodies, suggesting that it will be more immunogenic in the clinic. All of our low immunogenic biotherapeutic had lower percentage of positive donors, ranging here from 0 to about 21%, while our highly immunogenic biotherapeutic had about 58% positive donors. While a readout of T-cell proliferation to assess immunogenicity risk of biotherapeutics was very helpful, and as you can see, we found good correlation between predicted and the clinical ADA. We have decided to use a different assay and develop a different assay to look at the T-cell response towards biotherapeutics. In this assay, we minimize the time that's needed for the readout. We are using a short assay that looks at early markers of the T-cells, CD134 and CD137. The unique thing of this assay is that it doesn't require the use of both markers to be expressed. But if either of the markers is expressed, we will call this an activated cell. This reduced our time for a panel of 40 donors from 15 weeks to 2 weeks. It reduced our labor and cost by 80%. So how does this assay work? We use frozen PBMCs, culture them with the drug for 48 hours, and then by looking at flow cytometry, we gauge on the CD4 live population and the markers of CD134 and or CD137. We calculate the stimulation index. This is the treated versus untreated of expression of those markers. And by statistically looking at different molecules, we have found that SI over 1.8, meaning that activation of those markers is 1.8 than the untreated cells. We will call this a positive donor. This assay is currently used as part of the pipeline of Genentech to assess immunogenicity risk of biotherapeutics. Here is an example of how we use the T-cell activation assay to assess the immunogenicity risk of multiple biotherapeutics. In this case study, I used the same molecules as we used for the proliferation assay. To remind you, bevacizumab is the low immunogenic Genentech molecule. RG7652, avolucumab, alirucumab, and bococizumab are all anti-PCSK9 antibodies. But while bococizumab had 48% clinical ADA, all the rest were shown to have low clinical ADA. So we use this assay that I just described to assess how they will activate those biotherapeutic, the T cells, in vitro. You can see here that following the stimulation of bococizumab, half of our donors had high T cell activation in their stimulation index. If we look at panel B, for example, when we plot it to a table, to a graph, you can see here that bococizumab had much more positive donors than all the rest of the anti-PCSK9 and much higher than bevacizumab, suggesting that bococizumab will have high ADA in the clinic. Here is another panel that we have examined with the T-cell activation assay. We have used different molecules here that targets anti-IL-17, ixekizumab and sacakinumab. You can see here that ixekizumab has shown in the clinic to be more immunogenic than sacakinumab. It had 22% clinical ADA, while sacakinumab had lower than 1%. So how is our assay performed? In this slide, you can see our results of the evaluation of ixekizumab and sacakinumab. You can see here on the y-axis, this is the stimulation index, how well the cells the CD4 cells got activated. You can see here that following the treatment of PBMCs with ixekizumab, 13 donors out of the 40 were positive, had a stimulation index above 1.8 for their CD134 and or CD137. While following stimulation with sacakinumab, there was only one donor positive, suggesting that ixekizumab will be more immunogenic than sacakinumab when we will enter the clinic. Since those assays are trying to assess what will be the immunogenicity risk when we will administer this biotherapeutic during clinical trials or later on when we market, we are trying to capture as much diversity of the HLA class II as of our population that will require the biotherapeutic. Typically, in a T-cell proliferation or activation assay, we would use 40 or 50 different donors or PBMCs for us to get a different diversity of the HLA class II. And by that, hopefully, we will get the same frequency of different HLA class II as our population. You can see here in the slide, this is a few different HLA common they are in the worldwide population versus our T-cell assay. It is also worth mentioning that if we know of a specific population that we target the biotherapeutic to it, for example, in different areas, we might have a different diversity of the HLA class II. We may want to enrich this in our study, in our assay in vitro. In this slide, you can see a summary of the clinical ADA frequency and the percentage of positive responding donors in our T-cell activation assay. Our y-axis is our clinical ADA. On the x-axis, you can see the percent of positive donors in our in vitro assay. You can see here in the bottom of the slide where the low ADA biotherapeutics are that we were able to predict or assess their immunogenicity risk in their T-cell activation assay. You can see that we had very low percentage of positive donors. On the red, you can see the molecules that had intermediate or high ADA in the clinic and how those perform in our assay. You can see that for most of them, we had a good percentage of positive donors responding, meaning a very strong correlation between our predictive and clinical ADA. We also had here, you can see on the yellow mark, this molecule I. This is Humira. Humira we weren't able to assess the immunogenicity risk of Humira in our in vitro T-cell activation assay. This could be due to the MOA of Humira that may interfere with our assay readout, as it is a TNF alpha inhibitor. Therefore, we always need to think about the MOA of the molecule when we are running those assays and to find the best fit of assay to our molecule of interest. Another example where we need to think about the MOA of the molecule and then choose the proper assay or immunogenicity in vitro assay is, for example, for molecules that activate T cells. For those molecules, such as TDBs, for example, we want to know if the activation of the T cells is due to the immunogenicity of the molecule or due to the just MOA. The molecule is just doing what it's supposed to do, activating T cells. For those type of molecules, we will prefer using a DC T cell assay. In this assay, on day 0, we isolate PBMCs and isolate CD14 monocyte cells. Those monocyte cells, we will culture them with different cytokines, leading for differentiation of those monocytes to a monocytes-derived disease. We will mature those DCs and add the biotherapeutic. At this step, we expect that the biotherapeutic will be internalized by the DCs and start being presented by the HLA class 2. We will wash thoroughly the drug out of the culture. Then our DCs won't have the drug in their culture, but only DCs that are expressing the peptide. If this peptide is immunogenic, the peptide of the biotherapeutic, if it is immunogenic, will lead to an immune response. At this day 7, we isolate CD4 positive cells out of PBMCs and then culture them with those DCs that we have stimulated with the drug and currently may or may not express different peptides of the biotherapeutic. Then we can evaluate the T cell response by looking at T cell proliferation, cytokine secretion, or T cell activation. And then by looking at those different outputs, we can conclude if a molecule is immunogenic or if it's not. And then, since the CD4 T cells will never see the drug, we know that for sure this is not due to the MOA of the molecule. In summary, immunogenicity induced by biotherapeutic can lead to detrimental consequences on drug development and to patients' life. There are multiple factors that can influence immunogenicity, and we need to consider all the different factors there when we assess immunogenicity risk and give a prediction. In vitro assays can be used for candidate selection, for example. If we have multiple candidates, we can select the best candidate that lead to a less immune response, and also for immunogenicity characterization. Later on, understanding what caused the immunogenicity of a specific molecule and thinking if we can de-immunize, remove those areas that led to immunogenicity, generating a second generation of the biotherapeutic without an immunogenicity risk. However, we always need to consider the fact that those assays are assessment. They assess the risk of immunogenicity. The true ADA percentage can only be based upon the clinical data. Before finishing my talk, I would like to thank the different members of the Bioanalytical Science Department in Genentech, specifically to Patty Singonza, Shan Cheng, and Steve Swanson for their support. To the different members of the Predictive Science Group that have generated the data that I showed you today. We are always looking for motivated scientists to join our team. Please reach out to me if you are interested to learn more and maybe join the immunogenicity prediction group within Genentech. And now, let me pass the mic to Andres that will continue the discussion on immunogenicity.
So hello also from my side. My name is Andreas Radek, and I will present you today how we from Miltenyi Biotec support you for primary cell isolation for predictive immunogenicity. After a short introduction, I will come to our ideal cell separation solutions and our automated high-quality cell separation devices. And at the end, I will give you an example for a specific automated primary cell isolation. You heard from a previous speaker already the importance of immunogenicity prediction. I would like to highlight here the importance about the primary cells, either patient-derived or healthy donor, to use them for in vitro assays for predictive and immunogenicity studies. As you all know, both innate and adaptive immune cells play an important role in anti-drug antibody generation, and therefore assays using primary cell source can display a physiological relevance. There are more benefits of such cell types, such as these allow assays to be run in higher throughput and in miniature version, which can help us to assess potential immunogenicity as early as during the drug development process. Since immunogenicity response heavily depend on patient population, using primary cells from different HLA donors can give better estimation on the response in large patient population. Of course, such assay also allow us to zoom within cell-specific response. Within this presentation, I will also focus slightly on the MAPS assay and how we from Miltenyi Biotec can support you therein. For the MAPS assay, primary cells as the monocytes can be directly isolated with different automated cell separation devices from Miltenyi, for example from Buffycode. And we also support you for the first initial analysis of this starting material with our MAX Quant analyzers and with our 8-color immunophenotyping kit to analyze all the cell populations in the starting material already. After cell isolation, the monocytes have to be differentiated to immunogenic cells in the presence of cytokines, and these premium-grade cytokines are also provided by Miltenyi Biotec. The differentiation can be monitored using our MAX Quant analyzer. Then the dendritic cells are loaded with the model monoclonal antibody and induced to maturation with lipopolysaccharide. After 24 hours, major DCs are frozen, and after lysis, the HLA-DR peptide complexes are isolated via immunoprecipitation using anti-HLA-DR-coated beads. Finally, after several wash steps, the peptides are eluted from the HLA-DR complexes by a pH shift and then analyzed by nano-LCMS with subsequent sequence identification using database search. And now I will present you our MAX technology, which requires only 3 easy steps. So the first step is the magnetic labeling, where an antibody conjugated to a tiny paramagnetic microbead binds to a receptor of a protein on the cell surface. The next step is the magnetic separation, where the cells are passed through a column that is positioned within a magnet. The cells labeled with the microbeads are retained in the column, while everything else is washed through in a gentle way. The flow-through is called the negative fraction, containing the cells without the ligand for antibody or antibodies that were used. At the end, there is the elution step. The column is removed from the magnet, enabling the magnetically labeled cells to be washed out. This can be done either manually with a plunger to push the cells out, or automated using a vacuum to pull the cells out, or increasing the flow speed of the buffer in the system. All 3 methods are gentle for the cells and separate the cells into the negative and the positive fraction with exceptional purity and high viability. The MAX technology is therefore based on 3 components. The first one is the microbead, the second one is the column, and the third one is the magnetic separator. So why do we use MAX columns? In general, the magnetic separators in column-based and column-free systems have a comparable magnetic force. The difference between the systems is the column that is filled with little iron spheres. Each iron sphere passes a magnetic force onto the next over a very small distance, amplifying the magnetic force by 10,000-fold, ensuring that the magnetic field is constant throughout the column. Thus, minimal labeling with cells with tiny nanobeads suffices to retain target cells in the column. The iron spheres have a cell-friendly coating. Moreover, cells can run freely through the column and can be washed thoroughly to remove cell debris and contaminants. All these contributes to the fact that MAX columns allow the cell separation with minimal effects on cells And therefore, MACS columns are the best choice for cell separation with minimal effect on cells for translational applications. And now I will show you our solutions for automated high-quality cell separation. We have 3 devices for automated cell separation. You see on the lower right the AutoMACS Pro cell separator, which is a fully automated cell separation device for up to 6 samples. And then we have the MultiMACS X-Selected Separator Plus, where you can separate up to 24 samples in a semi-automated way. And this MultiMACS X-Selected is integrated into the MultiMACS X, what you see in the upper part. And I will come into detail to this device later in my talk. But now I want to show you how automation improves the efficiency in everyday cell processing, because less hands-on time allows you to work on more important tasks in the lab and also in the office. By the way, the reproducibility is also increased because no user variability and no human errors occur during a run over the full process time. The standardization is also one key factor for automation because you run always the same protocol, you get the same results and the same performance every time. And last but not least, one key factor is also the safety, because it reduces contaminations and employees' exposure to samples. So in summary, you run fewer experiments and fewer redundant experiments and focus your time and energy where it matters. As I said in the following slides, I will focus on the MultiMACS X as our fully automated cell separation device for highest throughput. With the MultiMACS X, it's possible to label and separate cells in a fully automated way. Due to the fact that the MultiMACS X-Selected is integrated into a liquid handling robotic system, the robot features to pipette samples, reagents, buffer, and cells during a fully automated process. And the integrated gripper arm allows us to transfer column blocks, trays, and pipette boxes on the work table, on the magnet, and as well into the storage hotels. The integrated elution chamber from the MultiMACS X-Selected enables also an elution of the cells from the MACS columns with a vacuum impulse. With the MultiMACS X, it's possible to isolate primary cells from different starting material. From human samples, you can either use PBMC to directly isolate your cells from this cell source, as well as directly from blood sources like whole blood, Buffy-coat, or leukopec. And we can also process marine samples from dissociated tissue on the MultiMACS X with up to 24 samples per run. The sample volume ranges between 100 microliter as the lowest volume and up to 200 milliliter per run, for example, for a full leukopec. For the MultiMACS X, we have also a customized application service established where we adapt your process onto a fully automated process on the MultiMACS X. As well, we have also established a set of base applications, which I will present you in the next slide. The base applications are designed to be ready for standard cell separations using the MultiMACS X. One of the base applications is the isolation of PBMCs directly from different sizes from leukopec in a fully automated way. We have also established the phycol-free cell separation directly from whole blood, Buffy-coat, and leukopec using our straight-from portfolio. Based on PBMC, we have 2 different base application sets. For example, for isolation kits, which is an untouched cell isolation of various cell types directly from PBMC. And the microbeads base applications are positive separations of specific cell types from PBMC with nano-sized microbeads. In the next slides, I will focus on selected base applications for primary cell separation, and I will start with the straight from whole blood application. Straight from whole blood again is directly isolation from the blood source, and you can run up to 50 milliliters of whole blood sample per donor with up to 3 donors in one run in parallel with up to 105 minutes process time depending how many donors and what sample volume you have selected during the process. And I've shown here the results of the testing of CD4 isolation of T cells and CD14 monocyte isolation. And you see in orange the high purity what we get out of the base replication separation with the MultiMACS X. In the next graphic, I show you the straight from Buffycoats example, and there you can either run 40 milliliter or 80 milliliter of Buffycoat sample per donor, and also up to 2 donors are possible to run in parallel in one automated process with process time between 48 to 70 minutes. Again, is here shown the CD4 and CD14 marker, and you see again a high purity above 90% for both selected cell types. Last but not least, I will show you the results of the isolation kit-based application for primary cell separation. And here you can run either 1 to 4 PBMC samples per run in time between 50 to 60 minutes for the full automated process. And I selected here the results from different kits, for example, the CD4 T cell kit, as well as the PALM T cell kit for separation of all T cells, as well as the isolation kit for PAN monocytes. And you see in the lower part that other kits are also established as base applications, what you can use with the MultiMACS X for different research topics. After the base applications, I come now to an example for customized application service, what we have for the MultiMACS X. So we develop here a tailor-made application for primary cell isolation for predictive immunogenicity assays. In this example, we use the positive separation for CD14 positive monocytes from PBMC in highest throughput, so from up to 6 to 8 PBMC samples, for example, generated from Buffycoat, in a parallel run on the MultiMACS X using CD14 microbeads over one multi-24 column block. After that, the non-labeled cells, so the flow-through, the negative fraction is collected, and from this flow-through, the CD4 positive T cells from the same donor are isolated untouched with an isolation kit to have at the end both cell types, so the monocytes and the T cells derived from the same donor, to do co-cultivation of both primary cells, first for model-seed generation of the monocytes, and then for the MAPS assay or for DC T cell assays. And at the end of my presentation, I want to summarize for you how we from Miltenyi Biotec support you to enhance your standardization for predictive immunogenicity assays. So I've shown you some examples for primary cell isolation with the MultiMACS X, for fully automated cell isolation with flexible reagent selection. But we have also cycle-free PBMC isolation established to generate PBMC donor banks from different starting material. And what I've shown you here is also for the immunogenicity, the isolation of monocyte and T cells for MAPS assay, but also for dendritic cell T cell assays from the same donor. Additionally, I've shown you our input from the cell culture portfolio. So we have recombinant cytokines for highest cell culture reproducibility, and the modices can be generated using our protocols with premium-grade cytokines. Last but not least, we have a high portfolio of flow analysis. We have a high throughput flow cytometer and recombinant antibodies to have a standardized quality control, for example, for dendritic cell phenotyping in the process of generating of major disease, to high parameter assay readout at the end. And finally, I want to say thank you for your attention.